Protein Dynamic Solutions, continues its efforts to provide the advantages of our system to the biopharma industry through a series of videos demonstrating the capabilities of our Protein Mentor platform technology. Protein Mentor is one of the few key analytical tools offering comprehensive and rapid screening methods for developability and end-to-end comparability assessments. A transformative analytical tool that delivers a 21 CFR Part 11 compliant platform method for implementation across the entire drug lifecycle, enabling process optimizations. The drug development process, which on average takes 10 years or more, begins with the exploratory phase, where scientists determine a disease pathway and identify potential targets for drug design. It is then followed by preclinical development, during which extended characterization is required. Protein Mentor satisfies this need by providing streamlined workflows requiring minimal sample preparation and only 1 microliter of sample per well, providing a comprehensive, direct, multi-attribute assessment of the intact protein in its formulation to de-risk preclinical candidate selection. These workflows make use of a proprietary slide cell array that can be subjected to accurate thermal control. Here are 3 examples demonstrating the flexibility in the design of experiments using the slide cell array. The first is a row setup for up to 3 candidates, and the second is a column setup for up to 7 candidates. Both setups contain samples to explore the physical-chemical properties such as concentration, pH, thermal incubation periods, or varying excipients for formulation optimization. The third is for comparability assessment of up to 21 candidates, ideal for developability assessment and lead preclinical candidate selection. All these approaches yield comparable data for the determination of the critical quality attributes, even under high concentrations and high dosing conditions required for subcutaneous administration. The comprehensive nature of the evaluations and the large data sets that can be gathered in record time are of high quality to enable deep machine learning, address concerns, and provide predictive results for decision-making. That same data is also used during the pilot scale phase to validate processes and propose pass or fail parameters, after which the technology transfer documents are drafted to commence manufacturing, allowing for our platform method to be transferred as well. Batch-to-batch comparisons are performed throughout the manufacturing phase to monitor the drug substance using the flexible design of experiments that take advantage of 2 different slide cell array setups. The first is a row setup for up to 3 candidates in septuplicate, and the second is a column setup for up to 7 candidates in triplicate. The last evaluation, which takes place during release testing, utilizes the same 2 slide cell array setups to evaluate the drug product before sending it to the final stage of storage. Guidelines, such as the International Council for Harmonization Q5E, on comparability of biotechnological or biological products subject to changes in their manufacturing process, can be effectively implemented using our platform method, which addresses the challenge of providing accurate, reproducible, and statistically robust results for an end-to-end comparability assessment. The goal of the comparability exercise discussed in the guidelines is to ensure that process changes have no adverse impact on the regulated drug product, and to assess the impact on product quality attribute differences to ensure improved product quality, safety, and efficacy. The comparability strategy is designed to determine risks during release testing, extended characterization, process control, and implemented during updates of a control strategy. Our platform method is ideal for establishing process control changes and examining deviations within the manufacturing process in just 10 minutes, with a side-by-side comparability assessment for up to 6 candidates in triplicate, against a reference standard at a constant 25 degrees Celsius. A fit-for-purpose quality by design, enabling reporting for end-to-end comparability assessment. The results ensure the International Council for Harmonization Q5E guidelines are being followed to design an optimal bioprocess, with an appropriate risk mitigation strategy. Implementing Protein Mentor's platform method throughout the drug life cycle will provide the flexibility to thoroughly investigate multiple attributes of preclinical candidates during the developability assessment and serve as a robust and rapid solution for end-to-end comparability assessments while accelerating speed to market.